So specific, the treatment is first, IMVOKE. Talk about the IMVOKE. IMVOKE is special shells in the California Sea. The blood of the Megathura and crenulata will be extracted, cleansed, and purified, then passing through the GMP facilities, and then it will be well dried and it made as a product called IMMUCOTHELs. So what is the role of the IMMUCOTHEL? As I mentioned before, cancer cells have a specific antigen called T antigen, TF antigen. Our T cell cannot properly recognize. So what should be done? IMMUCOTHEL has lots of T antigen. So once IMMUCOTHEL will be injected to our body will produce TF antibody. Then the antibody will attack the antigen on the cancer cells. So antigen-antibody reaction will take place. So what is the result? The tumor can be lysis, dissolved, destroyed. This is the major role of the IMMUCOTHEL. This is called specific immune stimulation. How about other non-specific immune stimulation? Like NK cells, the B cells, and T cells can be activated. That's why IMMUCOTHEL is called immune stimulant. We study about the selenase again. As I mentioned, cancer cells has high glutathione concentration. That's why cancer cells very strong. Chemotherapy cannot destroy easily the cancer cells. But once the high dose sodium selenite will be given to the cancer cells, then this selenium can enter very easily into the cancer cells. The selenium will bind 2 moles of glutathione. So the selenium make SDG. SDG means seleno-D-glutathione. Once SDG will be produced, the concentration of the glutathione will be decreased. Okay, that's why the barrier or the protection power of cancer will be lowered. So the chemotherapy can easily attack the cancer cells. Not only this SDG will produce ROS. ROS is reactive oxygen species. Once ROS will be produced inside the cancer cells, then what? Cancer cell cannot survive. Apoptosis can take place very easily. So the apoptotic body can be seen. The highest dose selenium can be considered as a targeted chemotherapy. Is it really reasonable mechanism or no? This mechanism was produced by the Dr. Michael Werenstedt. He is the professor of the Karolinska Institute, Sweden. Karolinska Institute is number 6 in the world, top class medical school. Not only that, this university is announced every year Nobel Prize laureate. The authority of this university cannot be negligible. It's very high. Not only that, selenium was first discovered in the world 200 years ago by Dr. Berzelius. The study of the selenium is very specific. It's really respected in this university. That's why we just accept without any questions. So the more specific mechanism like this, a little bit difficult, but maybe you will understand well. This is cancer cells. To produce more glutathione, it needs raw material. The raw material is cysteine. The cancer should attract or collect cysteine into the cancer cells. How? This is the XC antiporter. Through XC antiporter, it accepts the cysteine. Once cysteine will be entered into the cancer cell, the cysteine can be easily reduced to cysteine. Then what happened to our cysteine? Cysteine is from cysteine. This is some kind of tumor microenvironment to protect the cancers. Cysteine will be oxidized to cysteine. The cysteine will be sacrificed itself to cysteine. Then cysteine can easily enter the cancer cells. It is raw material for the cysteine, and then the cysteine will be produce lots of glutathione. You can get my point. This is the mechanism of how increase the glutathione into the cancer cells. So what happened here? Once the cysteine can be easily oxidized to cysteine, then if we inject the sodium selenite with 3 molecules of oxygen, then this will be easily reduced to HSE, selenite. So selenite is very light. Selenite is very heavy with 3 molecules of oxygen. This one cannot enter easily, but this one can enter into the cancer cell very well. You can get my point? That's why it can be accumulated inside the tumor cells 10 to 13 times higher than normal cells. So it is selectively accumulated inside the cancer cells. So from now on, we study our hypothermia. We have special hypothermia in IMVOKE system, 4 kinds of hypothermia. This is some kind of local hypothermia, neothermos, and this is remission 1, local also hypothermia. The 2 is systemic. This is PBM, photobiomodulation, using the NIR light. This is salt therapy. Also, this hypothermia. We'll discuss one by one. Local hypothermia first. It's selectively applying high-frequency electric current to the tumor area to create electric field. Then the electric field will generate heat. Then the cancer cell is very sensitive to heat, especially in a little bit higher temperature. The oxygen supply will be blocked and proliferation will be inhibited. Also electrolyte balance around the cancer cell will be disrupted. This leads to the destruction of cancer cells membrane and ultimately resulting in the death of cancer cells. This is the mechanism of hypothermia. As mentioned, this is neothermos, and this is the remission 1. The 13.56 of the local thermia megahertz, this is for the neothermos, 0.46 is for remission 1, local hypothermia. We'll study our salt hypothermia. Our salt is not ordinary. It's very, very specially made. How? General salt is only 50% of the pure salt. What else? Contaminant? Bitter magnesium chloride, almost 30%. Toxic. And then gas, 10%, and other mineral, 5%. This is a natural or general salt. It should be what? Melted above 1000 degrees by centigrade. Then what will happen? Moisture and gas will evaporate. But still, the mineral is remained. So what should be done? At least 15 days should be needed to remove all the minerals. Then pure crystal salt will be produced. That is for edible powder. But our salt hypothermia, not the 15 days, only 2 days. Only 2 days, and they will be made this way, black, salt black. This is our salt hypothermia raw material. This slide shows the edible salt, pure salt, 100%, no mineral is contaminated. So it is crystal salt. What is the benefit? Usually some people worry about blood pressure. If you enter the hot atmosphere, oh, I'm the high blood patient, or I cannot enter this kind of hot area, but the blood pressure can be lowered, not increasing, because of this salt. Very special. The temperature is not so high, only 40 to 42, very low, but it reaches into our body deeply. It will dilate our blood vessel. It will perspire a lot of the sweat, and the heart rate also. Ordinary facilities is usually heart rate will be increased, but the salt hypothermia never increased, maintained almost the same. So don't worry about the blood patient. Another one, this area is very sterilized because this is salt. Salt evaporation is covered all these rooms. So any kind of germ, bacteria cannot survive inside this. After 12 hours, no germ can survive. It's very hygienic. What is the PBM? PBM is the photobiomodulation using the NIR. NIR is near infrared. It's really special, our treatment. What spectrum you are using? This is ultraviolet. This is visible ray. So this is the red ray. After the red ray, it's called infrared. But near one, this is near infrared. This area is far infrared. IMVOKE PBM is using 810 and 940. The wavelength is very important. Also, dose is also important. What is the mechanism of the PBM? Once NIR light was given to our body, it will activate the mitochondria, especially the cytochrome C oxidase will be activated. Then what will result? ATP will be produced. ATP is some kind of energy, so it will increase the cell's ability to fight infection and accelerating the healing process. Okay, ROS also manufactured, lots of NO will be produced. What's the role of the NO, nitric oxide? That is vasodilatation. The blood vessel will be dilated. So once blood vessel will be dilated, then it will increase the circulation and then decrease the inflammation. Also, it will transport an oxygen and other immune cells will be better in the transported. Also, lymphatic vessel also can be the good function to lymphatic drainage. Okay, this is the 2016 the Harvard study, angiogenesis, neural synapses formation, and then SOD formation, activation of NF-kappa B, also the stem cells regeneration. It's also enhanced lymphatic drainage, especially NO, nitric oxide will be released, increased blood flow. It is already well-known mechanism. So our specific PBM is 4 kind of the wavelengths, 810, 940, 1200, 620. It is covered with illite plate. Illite will generate energy, very special minerals. Not only that kind of mechanism, also once the light stimulated of the mitochondria, it reduced the blood glucose level also. How? Almost this is 5.5, this is 3.5, so almost 2 millimole per liter can be reduced. So what's the equivalent? Times 18 means almost 40. The blood glucose can be reduced, lowered, 40. It is amazing. So you buy the NIR. Well, not only the blood glucose level will be reduced, also the CO2, carbon dioxide, it will increase. So meaning to say it activates the metabolism. So from now on, we'll discuss very interesting topic regarding the NIR. This is not ordinary NIR. It can destroy cancer cells. Researchers published this article, scientists destroy 99% of cancer cells in lab using vibrating molecules. This is the membrane of the cancer cells. Once the positive ion will be attached to the cell membrane, especially the negative head, it will attach very closely, tightly. Then, by that time, NIR will be applied. It has energy. It will vibrate this complex, moving, vibrating. So, how many hours needed? Only 8 minutes. Only 8 minutes of exposing NIR. Then the cell membrane of cancer cell can be destroyed. Very amazing. They published this one. So this one is called molecular jackhammers eradicate cancer cells by bionic-driven actions. This is the researchers of the Rice University, Houston, Texas, last year. Very interesting articles. According to them, they are using the aminocyanine. Cyanide means, the blue. Later I'll explain it. Because of the molecular mechanical action, it will kill easily, very rapidly by necrosis. They are using the single photon. Also, the aminocyanine is not too high, very low concentration, and the low dose of light was very effective in vivo. Yeah, molecular jackhammer because of the jackhammer. So this is aminocyanine. Cyanide means, as I mentioned, it is dark blue. Because of dark blue, the NIR light can give the energy to the cyanide. It will accumulate the heat and it will start to vibrating. It's very important, the color, the blue. So this is the cell membrane of the cancer cells. And this is cell membrane of normal cells. What is the difference? The outer membrane of cancer cells is negatively charged, as you see. This is normal cells. Where is the negatively charged? Inside or outside? Yeah, inside. Inside is negatively charged. So this is the difference of the normal cell and cancer cells. So once outer membrane is charged with negative, so once the positive molecule will be attached here, it will bind together tightly. Okay, that is by the aminocyanin. Okay, that's why the destroy of the cell membrane can take place. Thus, this diagram shows this is bibronic mode. This is the frequency around 770 or 700, 670 or 600 LED intensity. They're using the 730, 630, 680, and 740. Yeah, they're using this LED. So what is the result? This is the mice with the tumor. NIR was given to it. So this is the result. The tumor size increased until 60 days. But after 6 days, it reduced the size. Oh, interesting. How about survival rate? Until 6 days, almost 100. After 6 days, a little bit lowered, but it can maintain almost 55 or 60. Survival was maintained. But how about others? Very low. It cannot survive after 60 days. Yeah, this is a quite big difference. So, how we can combine this PBM and IMMUCOTHEL? My strategy is, I suggest this looks like the pistol. Pistol should be loaded with a bullet. The trigger should be pulled, then the bullet can be exploded. But how about IMMUCOTHEL? Okay, once IMMUCOTHEL looks like the bullet, but if you inject at the acute point, then I think it is loaded pistol. How can be triggered? I think it is PBM, because PBM gives the power and energy. So this is a primo node. Primo node is what? This is a lymphatic vessel. Inside the lymphatic vessel, primo node is located inside, very thin. The size is only 20 nanometers, thinner than the hair. Through this line, the immune cells, and the hormone, stem cells, whatever, can transport it without diluting other lymphatic vessels, other the blood vessels. Okay, so this is a very unique system. That's why I prefer to inject IMMUCOTHEL in the acute point to reach the target tissue. So how can combine this theory and then PBM? This is IMMUCOTHEL. This is oxidized form, oxyhemocyanin. This is copper. That's why it's colored blue. This one, blue color, has the absorption wavelength is 347, 582.5, 625. It's very similar with 620. We already existing 620 nanometer LED in PBM. That's why I think because of this, IMMUCOTHEL was once injected to the acute point, then it will be activated. It will get the energy, the vibration can take place. So my conclusion is this one. It's enhancing the effect of IMMUCOTHEL through PBM. How? Inducing heat and vibration of IMMUCOTHEL on the wall of cancer cells. How? The positive charge of immunocyanin binds to the negative charge on the outer cell of the cancer cells. And then we can expect cancer cells destruction effect by the heat or vibration generated by immunocyanin absorbing NIR. What another mechanism we can expect? Absorbed NIR light energy will improve the production speed of ATP, meaning to say ATP can more fastly produced in mitochondria. It means activating metabolism. So the reaction and movement of the immune anti-cancer drug like selenase and IMMUCOTHEL can be activated. Yeah. So it will enhance the anti-cancer effect is expected as a result. This is my some kind of imagination. So what is our presentation conclusion? The sooner, the better for cancer patients. As I mentioned already, there is a better cytotoxic effect when using a high dose compared to the low dose. High dose is better. And then using combined therapy along with the high dose selenase, IMVOKE, and hyperthermia such as PBM, salt, local hyperthermia is more effective. Combined is better. So IMVOKE treatment is cost-effective therapy compared to general treatment. Maybe you think, oh, that treatment might cost very high. I don't think so. Compared to other immune therapy, it's really, really low. So please contact us. How it will be the price? Not so expensive. It's really reasonable. Thank you for this. I will conclude our presentation today. Thank you.